residence in Maastricht, if I am informed correctly. My name is Gunther Kenis, and it's my job to entertain you for the next hour or so with a subject that for some of you might not seem to be that interesting. Can you understand me? Yeah. Maybe not that interesting because you're not that familiar with psychiatric or psychological disorders. I mean, you are a molecular life scientist, and it might seem strange for some of you that indeed molecular life sciences has a lot to do with psychiatry. And well, I will try to convince you of this in the next hour. So we will mainly be talking about the neurobiology of some psychiatric disorders, but with a focus, of course, on genetics, genetic predisposition, and also how these genetic factors interact with environmental factors to predispose persons to develop psychiatric disorders. Maybe you're not aware of this, but brain disorders in general have the highest level of social and economic burden in the whole world. So if you talk about brain disorders, well, there's a group called neurological disorders, where amongst others you see there the neurodegenerative disorders like Alzheimer's disease and Parkinson's disease, which have a clear component of degeneration of certain parts of the brain. But also other disorders where there is something wrong with either connection between several brain structures, and that leads to very clear phenotypes like epilepsy, certain pain syndromes, also some autoimmune disorders like multiple sclerosis. But if I'm right, you have heard about that in other lectures. And there is a huge group of psychiatric disorders where a lot of people suffer from without even that it's really noticeable, but it really can cause some kind of social burden, maybe for the person himself or herself, but also for the environment around that person. And then we're talking about major depression and anxiety disorders. Most of you are familiar with that in one way or the other. Maybe you have some relatives and so on. Less known is schizophrenia and bipolar disorder, but they have a quite high prevalence. Does anybody know the prevalence of schizophrenia, for example? What would you expect? Make a guess. You're a scientist, you do not do any guessing, but just make a guess. What do you think? 1 in 10,000? In fact, it's 1 in 100. So depending on how you define the psychosis phenotype and whether or not a patient will develop the real full-blown schizophrenia, the figures are between 0.75 to 1.5, even 2% of the population that will suffer once in their life from either psychosis disorders or will develop bipolar disorder or schizophrenia. So it's quite a high prevalence. And then of course you have other syndromes like autism, post-traumatic stress disorder, ADHD, a lot to do with that, about that these days, and some addiction and personality problems. So what I just want to say is that mental illnesses have a huge burden, and it is therefore necessary that we also invest in this and to try to figure out what causes these diseases and how we can treat them. And so these are some figures from the WHO indicating that if you look at the years that are lost due to disability of a person suffering from a mental illness, then major depression, amongst others, ranks the highest. So it's quite a huge burden, and together with all other psychiatric neurological disorders. And even in this figure, it's not a problem that is only in a minority of children or has to do with getting old. No, it is also with people in the prime of their life. As you can see, major depression has a huge burden compared to all other diseases in Western women. So it is quite important to investigate this. Now, as I said, we were going to talk about gene-environment interactions, and well, there is a lot of environmental risk factors known for a lot of these psychiatric disorders. So these are all the psychiatric disorders in this top 15. But as you can see, summarized here some environmental risk factors that are related to the onset of many psychiatric disorders. And you can see these factors can occur during the whole lifetime. Even from conception on, we know that nutrition of the mother, but also some kind of stress factors, drug abuse by the mother, has a huge influence on the precipitation of, for example, ADHD, autism, and anxiety disorders, and also schizophrenia. Also, birth complications or obstetric complications have is a huge risk factor for mainly schizophrenia, and then all factors during development, early life, and also in later life, psychosocial factors, what we call life events happening in later life, and certainly also lifestyle factors are all known to have a risk to develop one or the other psychiatric disorder. Now, in the context of the genetic factors of schizophrenia, well, we will come to that later, but well, I think you're familiar with these concepts that are mentioned here, and we will see that, well, at least for schizophrenia, there's a lot of these factors have been found in a number of patients. So, to summarize, I will focus my talk mainly on schizophrenia as some kind of example, and if we have some time left, I will move to major depression. But the main risk factors for schizophrenia are in fact prenatal infection, hypoxia during birth, urbanicity, meaning growing up in an urban environment. If you grow up in an urban environment, you are 3 to 4 times as likely to develop schizophrenia as people that grew up in rural areas. We don't know why this is, and we also know that this is really a causal phenomenon. A lot of epidemiological studies have proved this, and we know that it's a causal factor, but we don't know what the underlying biological mechanisms are. Also, psychological trauma during childhood, like parents that die or being bullied, etc. These are common risk factors. Being a member of a minority group, so like immigrants, for example. And last but not least, the use of cannabis during adolescence has a huge impact on the development of schizophrenia, especially during adolescence. If you have never smoked cannabis until now, well, you're quite safe to start because the risk of schizophrenia will not be increased. It's usually when people are using this before the age of 16 that you have increased risk. So I'm not saying that you have to do this, although you're Maastricht, but I'm just saying that your risk to develop schizophrenia will be lowered, will be not as high as when you start using it in adolescence. So what is gene-environment interactions? I think you have heard about this concept before. So the question is, if you look at these risk factors, they are quite common. Like obstetric complications, well, they are not that common, but that happen quite often, at least some kind of mild oxygen deprivation. Childhood trauma happens quite often. Fortunately, not all of these people develop psychiatric disorders or even schizophrenia. But a minority of these people that have experienced these factors, they will develop the psychiatric disorders, and the question is why. And we know, of course, that your genetic makeup which also, of course, has an influence on the function of the brain, make people vulnerable to the effects of these factors to develop schizophrenia. So this is what we are going to look at. Now, I'm not sure what you all know of the brain. I know that if you have studied here in Maastricht, you have had one course, somewhere in year 2, where you have looked at the brain, and I'm not sure whether you have encountered this in detail later on. Anyway, so the basic cells of the brain is, of course, the neurons and the glia cells. What is important about the neurons, just a few features to remember, is that neurons, they connect to each other with very complex dendritic structures on their cell bodies. And usually they have one axon that extends and projects to other areas in the brain. And also there is a huge axonic complex that can interact with a lot of dendritic structures on the receptive neuron. Yeah. So what is important to know is that the complexity of this dendrite structure is really important. The plasticity of these dendrites and synapses that are formed here, these are very important in all aspects of the functioning of the brain, like learning and memory processes, but also in all the processes, for example, related to emotional regulation. And how we process stimuli from the environment, we need to constantly update this dendritic structure. So this is a very important aspect, and of course, you can already guess that the genes that are playing a role in the formation of these dendrites and formation of these synapses, they are also very important in predisposing people to psychiatric disorders. And then, of course, we know that the brain is not just a collection of neurons. There are several substructures in the brain. Well, I'm not going to go in detail in all of them. We will see them throughout the lecture. But as you know, we have the huge cortical areas, and then the subcortical structures, of which the main important one is the limbic system, if you should remember that, which amongst others is composed by the hippocampus, the amygdala, hypothalamus, and the entorhinal cortex, and it's mainly involved in emotion regulation. Now, as I said, that these sometimes the neurons can extend very widely over the brain. Then we usually are talking about the monoaminergic neurons that produce, for example, serotonin, dopamine, and noradrenaline. And these cell bodies lie in the brainstem, and they project to all these areas. This is more or less shown here, for example, so that we have from the brainstem several nuclei that project to all these different substructures and continuously modulating their function. And as you may know, these monoaminergic cell bodies have been implicated in psychiatric disorders for a long, long time. And that is how also, well, it was due to the discovery of the working mechanism of some antidepressants and antipsychotics that have really changed the functioning of the dopaminergic and the serotonergic system, that people were looking for this and they said, okay, there is something wrong with serotonin in depression and with dopamine in schizophrenia. However, this is of course an old view. It's still true, but there is a lot more going on. As long as you remember that it is about all these projecting neurons from the midbrain that have to connect to all these substructures, and this connection is really vital to have a normal functioning brain. This is another picture about this. So what we are looking for is in fact genes or genetic factors that disturb in one way or the other the cellular functioning. And when we talk about cellular functioning of a neuron, we talk about the neural activity, so how often or how easy it is for the neuron to generate an action potential. The plasticity, meaning how fast a neuron can react to its environment and form new dendrites, form new synapses. The signaling, so how well it receives signals from the surrounding neurons and also generates in this response an action potential, and how it connects to all the other neurons. This is all called the cellular function of these neurons. Well, if this is disturbed, of course you get either by genetic makeup or by the influence of environmental factors, you get changes in neural networks, which at this point we are now able to visualize these neural networks using functional MRI. Have you ever heard of this technique, fMRI? I will come to that later. Anyway, we are able to visualize the activity of certain networks in the brain. And of course, if you have these factors disturbing these networks, then it may lead to, well, psychiatric disorders, or it predisposes us to behave very differently in some situations from each other. Some people, for example, react to life's problems by increasing their intake of alcohol, while other people get depressed, or other people even see it as an opportunity to do something different. Okay, so why are we looking for genes in psychiatry? Well, unfortunately, we don't have that much insight into the disease, pathology, and etiology of psychiatric disorders, nor for depression, nor for schizophrenia, nor for the other disorders. We now begin to understand which networks at least are involved, but what it causes is very unclear. So by investigating these genetic factors, we hope to have some idea about the biological pathways that are involved in these diseases. And in that way, of course, we can try to develop better treatment strategies. But there is two other interesting things, and that is, one is that we are not really doing a good job in phenotyping patients or in making diagnosis of patients. So what is schizophrenia? What is depression? Psychiatrists nowadays, they use a set of kind of criteria, and if these are fulfilled, or maybe 4 out of 5 criteria are fulfilled, then we say, okay, this patient has a major depression, or this patient has an alcohol disorder. And psychiatrists base their choices of a treatment on these categories. But of course, they are very, should I say, they're not very strict criteria. So it's that's also why a lot of patients fail their therapy. In general, I think about 50 to 60% of patients respond to the first treatment for a psychiatric disorder. So if you're depressed and you go to a doctor, you get prescribed some SSRIs, some antidepressants, and only 50% of the patients really respond to this. And then you have to start a new treatment or combine it with psychotherapy and so on. And the main issue there is that we have no good diagnostic criteria. So once we know better these etiological and genetic pathways that underlie these diseases, we might incorporate this into the diagnosis. Maybe you have heard of this. There is now a new manual for psychiatric disorders, which is the Diagnostic and Statistical Manual for Psychiatric Disorders, in short DSM. The latest version is DSM-4, and now they are working on the new version DSM-5, where they are also trying to incorporate at least some biological factors to help the diagnosis and the treatment planning. But there's a lot of discussion about this. It's sometimes even on the news I've seen. So, well, at least genetic research can also take part in that. Another thing is that people do not just develop psychiatric disorder out of the blue. Usually, patients suffer from some disabilities for a while before they have or before they develop the real full-blown disease. We call this a prodromal phenotype, which we can measure by doing neuropsychological assessments, for example. But usually these people then function quite normally, or at least for the environment, it might be invisible. But they are at an increased risk to develop the disease. Sometimes these people present themselves to the clinic and they have some complaints, but it's too early to start a treatment. Treatments with, in psychiatric context, either with cognitive behavioral therapy or with antipsychotics or antidepressants. It has huge impact, so you don't do this when it's not necessary. So, but then it's up to the physician, to the psychiatrist, to try to identify the people that are really at risk to develop this disorder, or maybe they have just a temporal elevation of some of these symptoms. And we now know that also genetic factors may play a role in there. So, in fact, to monitor the transition from, well, a subclinical prodromal phenotype to a real full-blown disease. Anyway, let's have a look at what psychosis is. I'm not sure whether you're familiar with that, but in fact, psychosis consists of three main dimensions, diagnostic dimensions. One of it is the positive symptoms, so where people have either delusions or hallucinations. These are also the most known by people not in the field. So people, they are hearing voices, or they're seeing people that are not really there. Or they have some paranoia idea, so they think that they are prosecuted all the time. Things like that. This is the dimension that we call positive symptoms. And it's usually accompanied by a lot of negative symptoms, where you see a loss of emotional reactivity, no sense of reality, usually thought and cognitive disorder or thought disorder. So they're not clearly able to think, and they have some kind of flattened affect. In addition with this, and this is causing the real dysfunctioning of the patients, that is that they start social withdrawing, they have thought blocking, and also some well, real cognitive disorders that they are, for example, unable to plan any activity, etc. So these are the real disabling symptoms. So just for you to get an idea about what schizophrenia is, or psychosis. Now, how is psychosis caused, or what do we know now about the etiology of a disease? Well, we know that dopamine is very important there, because, as I said previously, antipsychotics, they reduce the dopaminergic neurotransmission. And if you take dopamine-stimulating drugs like amphetamines or even cannabis, for example, you may elicit some psychotic episodes. Now, you may remember that there are two different dopaminergic systems, and the one that we are talking about in the context of schizophrenia is the mesocortical limbic system, which is depicted here. So these dopaminergic cell bodies reside in the ventral tegmental area in the midbrain, and they project mainly to the prefrontal cortex, but also to the limbic system, and therefore, of course, the mesocortical limbic dopaminergic system. Now, what happens is that in normal states, these dopaminergic neurons in the midbrain, if you have some kind of stimuli from the outside, no matter what, your dopaminergic neurons, they react to this by giving a burst fire of dopamine, so they have a dopamine release in both limbic areas and the prefrontal areas. Now, this prefrontal cortex, in a lot of brain networks or psychological networks in the brain, the prefrontal cortex has an inhibitory activity. And so also here, so normally the prefrontal cortex gives negative feedback signal to the dopaminergic neurons in the midbrain and also to the limbic system. So whenever there is a stimulus, you get a spontaneous firing of dopamine. It stimulates the limbic areas. It stimulates the cortical areas, but the cortical areas gives a negative feedback, so to dampen this dopaminergic response again. Now, in the case of schizophrenia, what happens is that especially the functioning of this prefrontal cortex is decreased. So that whenever there is a dopaminergic stimulus, you get a high activation of the limbic areas, but a decreased activation of the prefrontal cortex, making it that the negative feedback to the limbic areas and the midbrain are hampered. So, now, the positive psychotic symptoms like delusions and hallucinations, as I thought previously, we think that they're arising from an increased dopamine stimulus in the limbic areas. And this is mainly the ventral striatum, nucleus accumbens, and hippocampal areas. While the prefrontal cortex is associated with these cognitive dysfunctions. Prefrontal cortex is very important in the mechanism of planning of actions, of thinking, working memory. All these executive functions, they depend heavily on the prefrontal cortex. So therefore, schizophrenia patients have a reduced dopamine functioning in the prefrontal cortex, leading to the negative and cognitive symptoms, and have an increased dopamine response in the limbic areas, causing positive symptoms, hallucinations, delusions. So this is about very basic, short, the neurobiology of schizophrenia, but you just have to remember it for a while so that you can follow the rest of my talk. Now, if you look at what causes schizophrenia, as I said, there are a lot of risk factors, as I told previously. Indeed, urbanicity and being a member of a minority population and the use of cannabis are of the highest risk factors. But of course, the relative risk ratio is still quite low. It's about maybe 2. So if you have experienced these, your chances is about 2 times higher that you will have disorder. But if you have a brother or sister or a mother or father with a disease, then you will see that your risk increases dramatically. So indicating that probably there are some genetic factors going on that increase your vulnerability to develop schizophrenia if you have a first-degree relative with schizophrenia. Now, as I said, a lot of people also encounter these environmental risk factors. For example, about 30% of the population lives in urban areas. At least in the Netherlands, 20% of people use cannabis before the age of 60. And well, trauma and serious life events, so high stressful factors in life. Well, all of us encounter this now in a while, but it's in combination. Why does it lead to the prevalence of only 1%? Of course, because you need to have this liability to develop schizophrenia. Now, so there must be some kind of genetic factors playing a role in schizophrenia. So how to find these factors? Well, the general idea for any complex disease to find the genetic factors is summarized here. So you're going to look in family studies whether there is some genetic component. So you make an estimation of the heritability, so the degree of familial aggregation. After that, you want to find the genes that are responsible, and you can do that in like linkage analysis. So also in family studies where you try to find a chromosomal location that is always inherited together with your disease. And once you find that, in that region, you try to find the responsible causal variation to develop the disease, and you do that in the case control studies or association studies. So I think you're familiar with the concepts of linkage and association. Yes? No? Some of you? Okay. So if you look at the heritability, well, heritability is a quite abstract construct. So it's about a proportion of the variation in the phenotype within the population that is attributed to genetic variation. So in fact, your phenotype, of course, is brought about by both genotype and environmental factors. And in fact, it's the contribution of the genotype that is some kind of factor called heritability. And you can estimate heritability using, for example, also twin studies. In some cases, there is also some relationship between the genotype and the environment. Do you know how that is called? It's a very important concept. It's called gene-environment correlation. So do not confuse this with gene-environment interaction, because it's something completely different. A typical example in the case of behavioral analysis would be that people with a certain genetic makeup or with certain genotypes, they are more likely to take higher levels of risks. And so they're going skydiving and things like that. So they have a real drive for novelty seeking and taking high levels of risk. And this is partly due to a genetic makeup. So of course, these people, they also have a higher incidence or higher risk to be involved in accidents. Right? If you're skydiving, there is a reasonable chance that you can have some accidents, or at least break a leg or something. If you do not do skydiving, your chances are lower, of course. So based upon your genotype, you can put yourself in some kind of environmental factors more than other people. Now, if you look at the heritability of psychiatric disorders, it's quite high. So these have the highest heritability of all complex diseases. If you compare it to cardiovascular disease or diabetes or whatever, psychiatric disorders have the highest heritability, especially autism, but also psychotic disorders, bipolar disorder, schizophrenia, and also more affective disorders, so anxiety and major depression. Another way to put this is, as I told previously, if you look at the shared genetic background. So if you have a first-degree relative, you will have about 10% more chance to develop the disease. The general population, the occurrence is 1%, but if you are, for example, a monozygotic twin of a patient with schizophrenia, then your chances to develop disease is also 50%. So clearly indicating with these kind of population studies that that there is a genetic component in schizophrenia. Now people try to look for these causal genes using these linkage and association studies, so I do not have to explain you what these linkage analyses are. And this is a summary. It's already old, but there is nothing new there. That there have been a lot of chromosomal locations associated or linked to development of schizophrenia. I think this is a later slide. You can see that indeed a very substantial number of chromosomal loci are linked to schizophrenia. So that means that there are probably multiple genetic factors involved, and also that these factors may be population or even family dependent, right? So that you have, in some families, you have one variant causing the disease, and in other families you have another variant or another gene causing the disease. So if you combine that in a huge linkage analysis study, it's very hard to find consistent findings, right? And of course, there is also some overlap between diseases, especially between, for example, schizophrenia and bipolar disorder. And this, of course, comes back to the phenotyping. As I told you before, it's very difficult to make a distinction. For example, certainly in the early onset of schizophrenia, is this a full-blown schizophrenic patient, or is this some kind of psychotic episode of a bipolar disorder patient? So that's very difficult, and this is also shown in the underlying biology. So there is indeed some biological overlap between these disorders as reflected in the outcome of these meta-analyses. Nevertheless, there has been some attempts to perform these association studies, so to see whether some certain genetic variants, like single nucleotide polymorphisms or distributed differently between controls and patients. Well, there have been now, especially in schizophrenia, there have been huge studies combining all this. So in fact, what we try to do now is to combine all this data from linkage analysis and from association studies, but also, of course, from genome-wide association studies. And if I remember, if I'm correct, you have a lecture about this last week, about genome-wide association studies. Yeah, you remember? So in fact, what you do is you measure about 1 million single nucleotide polymorphisms at the same time, and you try to relate this to your disease, so whether or not they are more frequently in your patients or controls. So what we try to do in schizophrenia or in psychiatry is more or less the same concept as any other complex diseases. So using genome-wide association studies, combining these data with candidate regions from linkage analysis, and that gives us some clue about real genomic locations that are associated with the disease. So there is some kind of consensus of these risk-modifying genes, and you can find these back in publicly available databases. This is the one for schizophrenia, but there is also one for Alzheimer's disease, and even one for diabetes, if I'm correct. So there you can see the mostly associated genes of these diseases. And well, to wrap this up, or what has been found in these genome-wide association studies compared with this linkage analysis, that is that a lot of genes or genetic variants in genes that are involved in neural development, in outgrowth of dendrites and axons, and in synapse formation. So there are a lot of genes coming up of these association studies that all point to the formation of the connections between neurons. Some of them are really only expressed during development, so implicating that the developmental period where these factors are expressed are very important for the normal development of the brain. And if you have one of these environmental risk factors occurring at that moment, you're more likely to develop a disease if you have then such a genetic variant in this case. We see also that other genes are not only expressed during development, but for example, a process like synapse formation is a continuous process throughout life, so you need this continuous changes in the synapse. And environmental factors that impact on the genes important there can occur during any time of life and will lead to development of schizophrenia. Another group of genes that have been associated are related to neurotransmission, and especially dopaminergic and glutamatergic neurotransmission. This is a figure that explains this. If you look at all the data that comes out of these association studies, in the beginning it's fairly difficult to make some kind of consensus in there, because there are so many different genes associated with these diseases. But if you then combine this in a pathway, then you will see that a lot of these genes, they all have been implicated in schizophrenia, that they all have something to do with the outgrowth of these dendrites or of these axons. For example, the number one schizophrenia gene is DISC1. It's called disrupted schizophrenia 1 gene. This was discovered because a Scottish family really lacked this protein, and they all had full-blown schizophrenia, this family. But then we know that, or we now at least know that a lot of other molecules that interact with these genes or interact with the signaling of these DISC1 gene, they all can lead to psychiatric or to schizophrenia. Yeah. So it's not. It depends really on the family that you look in, or the population. In some populations, you have an increased frequency of a genetic variant, causal genetic variant in, for example, PDE4B, but in another population it might be a genetic factor in this one gene or even in some other signaling molecules. But it appears that whenever you interfere in this pathway genetically, that you increase the risk for getting schizophrenia. So this also indicates that indeed schizophrenia is some kind of neurological disorder or some kind of developmental disorder. So you really need a good development of the brain during early life. So both in uterus development, but also in the early life years. And secondly, that there is also, of course, these abnormalities in the neurotransmitter signaling. The neurotransmitter signaling, as depicted here, so we saw that there was a lot of genes coming up that disturb the glutamatergic signaling and dopaminergic signaling, as you can see here. But this glutamatergic signaling also really interacts with the development of these dendrites. Maybe you still remember from your earlier courses, if you want to make new connections, you need activity there. And you need some kind of, your brain cells have to be used, they need to generate action potentials, they need to receive action potentials, otherwise you will not form new synapses, so you have to use your brain. It's all the concept of learning, of course. So of course that these glutamatergic receptors, these glutamatergic signaling molecules that are so important in generation of these action potentials, they of course interact with all of these molecules that form synapses and form dendrites. So at least now we know that schizophrenia is caused, and this is proven from the genetic point of view with these association studies, that schizophrenia is caused by disturbance in the development of the brain. Now, if you first have a look at this slide, if you look at I summarized some of these risk factors earlier, and I said they can occur throughout life. So it's, for example, cannabis use, trauma, urbanicity, and being a member of a minority group. But we can see some kind of window where they're extremely important. And as you can see, it usually is just before adolescence. So early puberty, if you experience there some of these risk factors, then you have increased risk to affect key psychological and developmental processes that are important in schizophrenia. And I have summarized here some of these. It will lead us too far, but for example, what is very important in schizophrenia is a good development of social cognition. How you perceive the other people around you, and how to behave in a normal social context. So this is very important, and the development of this social cognition is also extremely active in this period. So if you have then some kind of these risk factors in this important time window, then it will disturb the social cognition, and it can lead to behavioral problems and might lead to schizophrenia. Now, what happens in this period, which is related to the genetic factors that I just showed you that have influenced the formation of synapses and so on? Well, that's shown here in this figure. Some of the prefrontal areas, your prefrontal cortex, is not mature when you're born. No. The huge development of the prefrontal cortex, or at least the functional development of this cortex, really takes place during adolescence. So this is where you can see this is at birth, and you can see how complex the prefrontal cortex becomes during adolescence. During adolescence, you have a lot of new connections that are formed. They're also explaining the sometimes aberrant behavior of adolescent boys and girls, because their prefrontal cortex is so much in development, and sometimes it just goes Anyway, not only, if you look at the molecular levels of this development of prefrontal cortex, then you can see that this has a lot to do with the formation of interneurons. You remember what interneurons are? These are inhibiting neurons, right? And they are the most important in regulating activity of different kinds of brain structures. So the prefrontal cortex receives inputs from all other areas, and it's the job of the interneurons to dampen all these responses. So a good development, functional development of the prefrontal cortex is accompanied with a good development of these interneurons. And they really develop during this adolescent period. Also, these dopaminergic projections that we talked about earlier, they are really quite in development in this period. And as you also see, the glutamatergic synapse density decreases. So when you're born, you have a lot of activating synapses, which is also good because you need to learn a lot. You need to take up all these stimuli and incorporate it and try to balance your behavior in correspondence with what you see in the environment. But during adolescence, you have what we call synaptic pruning. So a loss of glutamatergic synapses together with an increase in these regulatory dopaminergic synapses and an increase in inhibitory interneurons. So you can imagine that if you have a genetic variant that messes up this process, for example, this glutamatergic signaling, and if you have a disturbance in this glutamate receptor expression, and then if you really encounter one of these environmental risk factors in this period, you might interfere with the normal development of this process, leading to an abnormal development of your prefrontal cortex. And as I showed previously, the prefrontal cortex is very, very important in schizophrenia. Okay, is this clear to you? Does this make sense? If you do not understand, please interrupt me, because I have no idea about how good your knowledge of the brain still is, and how well you're able to reason in the context of these psychiatric disorders. Anyway, so this is more or less the summary. So all these developmental genes and neurotransmitter genes that are really important during the development of the brain during adolescence. If you disturb this, then you will have a higher chance to develop schizophrenia. Now, the question, of course, is, if you find such a genetic variant, and I already told you, they do not occur very frequent in the population. In some families, it's this variant, in some families, it's the other variant. That means that we cannot use this genetic variant as some kind of diagnostic marker, as some kind of tool to make some estimation about which people really will develop disorder and which people will not. So that's a problem. And people tried with these huge genome-wide association studies, try to find, well, the real risk variant that can explain at least a substantial proportion of variability in this phenotype of schizophrenia. And well, this is a summary. There was a huge issue in Nature where 3 of these large genome-wide association studies have been published together. In the meantime, there are many others that have appeared with all bigger and bigger well, more power, so more patients included. The problem only is that we know that these genetic variants have a very low odds ratio. So we're talking about odds ratio of maybe 1.1 to 1.2, and that's even 1.2 is quite high. So we know that these genetic risk variants do not explain that much, and you need, of course, a large power. You need a huge data set, and people have even estimated that you probably need more schizophrenia patients in these studies than there actually are in the world. Anyway, people are still trying to make something out of this. So you have seen these kinds of plots before. So this is what we call the Manhattan plots from the genome-wide association studies. Have you seen this before? Is this, no? So what you do in these genome-wide association studies, you have about 1 million variants that are measured, and you'll try to see how likely it is that they are differently distributed over your patients than over your controls. And what you in fact do is you plot the p-value of each SNP, and this is the chromosomal location. Now, if you do all these analysis, so you have to do 1 million statistical analysis. Of course, that gives you some statistical problems. With the type 2 errors, so you need some kind of correction. So your p-value will not be 0.05. With your p-value, well, there is now a consensus that for GWAS it should be 10 to the minus 8. In this case, well, this was 2 years ago, they still used 10 to the minus 7.5 or something. So in fact, what you can say, if you have any signal that is lower than this p-value, those really are significantly associated with schizophrenia. Now, first people saw that there was a lot of genes in the MHC complex. So MHC has a lot to do with the immune regulation. And well, this was heavily debated because people thought that this was some kind of artifact. And on the other hand, and what researchers are trying to do now is to combine not only just the stop genes or stop associations, but try to find a much larger portion of signals, maybe not 10 to the minus 7, but even 10 to the minus 5, and really try to see how these SNPs are commonly associated with disorders. So if you examine them separately, then you can see that at least a combination of these stop genes are always have an increased distribution or an increased frequency in schizophrenia and bipolar patients if you compare this to non-psychiatric populations. For example, cardiovascular disease, rheumatoid arthritis, and diabetes types. So meaning that indeed the genome-wide association studies do tell us something about well, the causal genetic variants and the causal biological pathways in these disorders. Although, as you can see here, the variance explained remains very, very low. So about some of them 3%, it's very, very low. But at least there is some idea that we can have from these genetic association studies about the causal pathways of schizophrenia. And if you then see they indeed relate to all these developmental genes. Now, of course, how then to explain the heritability of schizophrenia, which was estimated to be about 50%. That's quite low, and we only can explain 3% with all these common genetic factors. So that means where is this heritability? And this was discussed heavily a few years ago and still is. In this Nature paper, here the reference, it's quite, or they discussed this. And some factors that you also consider, because I know you're dealing with these genetic predisposition in this course, and you will see something about autoimmune disorders and so on. What you have to consider is that there may be a lot of hidden variants. So at this moment, we can measure even 2 million single nucleotide polymorphisms at the same time, so covering about 20% of the genome. But if you consider linkage disequilibrium between all these variants, we say that we can capture almost all the variation of the genome. Well, there may be still variants that we don't know, and that may have a huge impact. And that may not be linked to any other locus, which is not unlikely. Another issue is that we are only looking at common variants. Because we are looking at common diseases, we always think that these common diseases can be explained by common variants. We now know that there are multiple rare variants, and they happen quite often. But again, as I told previously, in some people it's other genetic variants than in other people. So it's very hard to find these variants if you just look at a huge cohort of different families and so on. And so these multiple rare variants, they may, if we could measure them, they could explain a lot more of the heritability. Also, in these genome-wide association studies, we do not consider copy number variants. And this is a very important aspect. I will come back to that later. What we also will measure is the concept of gene interactions. So it might be that a causal effect or an aberrant effect of one genetic variant is only visible in interaction with another causal variant. And if you just do these genome-wide association studies, you just test 1 million different associations and you not consider the interactions between them. So that also could explain why we do not find these high levels of heritability. And also, as I said previously, we are not very good, well, psychiatrists are not very good, I'm not a psychiatrist, but they are not very good in phenotyping patients. So we have a collection nowadays of, I think, about 50,000 schizophrenia patients, which we in Maastricht also take part of this. We deliver DNA of these patients. But what we call schizophrenia here in Maastricht is not schizophrenia in Estonia or whatever. So there is a huge discrepancy between the diagnosis of these patients. So that also might explain that we do not find these high levels of heritability. Then there is also, of course, the concept of epigenetics, what you will hear about next week. Epigenetics is all these methylation and histomodifications that are going on, and they're also partly heritable, but we do not measure them in these GWAS studies, so therefore cannot explain the heritability. And last but not least, there is the gene-environment interactions. So this is another interesting paper, quite dealing more conceptually and more statistically in this problem. So if you can see, it's not only for schizophrenia, by the way, also for other diseases like Crohn's disease and type 2 diabetes, we have a lot of loci identified that are related to these disorders, but they only explain a small proportion of the heritability. And as detailed in this article is, well, the reasons are again shown here. What we measure are these common polymorphisms. Right? And they all have a low effect size, a low penetrance, so a low disease effect. But diseases may be caused, even common diseases may be caused by rare variants that are quite rare, maybe have a low impact, but usually they have a higher impact. Well, in this case, we talk about Mendelian diseases. So when they have a very high impact, and then you also talking usually about diseases like type 1 diabetes or Huntington's disease and so on. Anyway, but we may be looking just at the wrong end of this graph. And as I said previously, discussed in a paper from the psychiatric GWAS consortium, that is that if you have multiple rare variants, they may increase your relative risk rather than looking for common variants in common diseases. Just one note about these copy number variations. I'm sure that you all heard about copy number variations. So it was only a few years ago that really it was discovered that a large portion of the genome is coming out of the 1000 genome project. A large portion of the genome is subject to large structural variation. So what we talked about now was things like single nucleotide polymorphisms, so a substitution of a single locus with two nucleotides. But it appears that large portions of the genomes are in fact duplicated or even deleted. And this is a figure of how often this happens, and you see it's quite a lot. It's even about 12% of the human genome is regularly copied or deleted, duplicated, or even triplicated, and so on. So, in fact, what you can see is if you look, for example, of the single intensity in the GWAS study, then you can see that, well, all variants have the same kind of intensity signal, and then there is a portion of somewhere in the chromosome where these signals are missing. Now, people also start looking for this in schizophrenia, and it appears that in schizophrenia patients there are much more de novo deletions than in normal controls. So these copy number variations, these deletions, they can occur, for example, during conception, during development, for example, of the gametes. It appears that in schizophrenia cases, these de novo mutations happen quite often. And if you look at the more common copy number variations, then we also can see that the genes that they disturb, these copy numbers, they are also mainly located in pathways that have something to do with brain development, again with these formation of axons and synapses. So again, pointing to the fact that genes important in axonal signaling and axonal growth, dendritic growth, are very important in schizophrenia. I'm going to skip this for a while. So, but one important issue in this missing heritability is that we do not consider the environment. Until now, we just focused on a genetic variant that may be causally related to the disease, for example, in genome-wide association studies, but they did not consider the effect of the environment. What if some of the people with the aberrant genetic variant did not have any of these environmental risk factors? Well, then they may have not developed a disease, right? But we also consider them as, well, a person carrying this risk factor. so it it gives a bias to this distribution between patients and controls in the genome-wide association studies. So again, the question is, why do people sometimes get a disease and why not? This is what I showed previously. So there is some kind of genetic component. You've seen that now also in the GWAS studies, but it's always caused by some kind of environmental factor. And let's focus now on one of the examples, which is the use of cannabis. We know that regardless of your genetic makeup, regardless whether you have genetic liability or familial liability to develop psychosis, if you use cannabis from an early age on, so this is in Dutch, it's a previous slide. Anyway, if you start using cannabis at a quite early age, you're much more likely to develop schizophrenia. But not all the people that really use cannabis will develop schizophrenia. This is one of the studies, also studies from here in Maastricht. Researchers did a lot of work on this here. So not only the age, but also the amount you use cannabis is associated with increased risk of psychosis. So more than 3 times a week, you increase your risk of getting psychosis. And it's also, as I told you, related to the age. So after puberty, your relative risk does not increase, or at least not that much, but it increases before puberty. And this has also been examined in animal models, and something that we also try to establish here. Now, if you look back to what I've told you previously, what are the biological causes of schizophrenia, or what is the neurobiology behind it, is this lack of dopamine processing in the prefrontal cortex. And this is a dopaminergic synapse, for example, in the prefrontal cortex. So dopamine synthesized out of tyrosine is released in the synaptic cleft and stimulates the receptors. Now, the most important regulator of the dopamine concentration in the synapse is not the release, but in fact how much it is broken down by this enzyme COMT. It's catechol-oxymethyl-transferase, so it breaks down dopamine, but only in the prefrontal cortex. In the striatum, for example, or in the midbrain, or in the little accumbens, it's not COMT that is the major determinant of dopamine concentration. There it is the activity of a transporter. So, but could it be that since this is only expressed, or mainly expressed in the prefrontal cortex, that a genetic variant in this gene may cause, well, disturbances in dopamine signaling in the prefrontal cortex? And indeed, there is some kind of polymorphism in this gene, leading to a substitution, a Valmet substitution at place 158 in the protein. So people that are carrying of the valine allele, they have a high activity of COMT, and people carrying the methionine allele have a low activity. So meaning that the Val allele carriers have a higher breakdown of dopamine, so there is less dopamine available in the synaptic cleft, while people with the methionine allele, they have higher levels of dopamine in the synaptic cleft. And what we have seen here, and together with people in the UK, that if you measure the genotype of cohorts of people with schizophrenia and without schizophrenia, and stratify them according to their use of cannabis, that it is this Val allele, so this high activity allele, that increases your risk to develop schizophrenia. So there was no statistically significant difference in the risk to develop schizophrenia, although it's lower here, but it's not significant, between these three genotypes. But if you use cannabis, then you're much more likely to develop schizophrenia if you're carrying these Val allele, or the double Val allele at least. So how can we explain this? Well, so the activity determines the dopamine response in the prefrontal cortex. So a high activity means low dopamine concentrations, and the activity in your prefrontal cortex decreases. Also, making that if you use cannabis, you're much more likely to have a disturbed dopamine response in your limbic system. As I showed this previous graph, prefrontal cortex gives a negative feedback signal to the midbrain and to the limbic areas. When this activity is decreased because of this disturbed dopamine breakdown, then you have an increased dopamine activity in the limbic system, leading to positive symptoms and the onset of psychosis. So this is a clear indication of these gene-environment interaction. So on its own, this genetic variant, this called Valmet allele, will not predispose you to develop schizophrenia. But in the context of cannabis use, so that you constantly are activating your dopaminergic system and signaling in the prefrontal cortex, then you will be more susceptible to the effects of cannabis and develop psychosis. Now, this is just an indication that there is something going on, but of course it does not explain what's going on at a real molecular level, because we now know that schizophrenia or psychosis may be caused by a hyperdopaminergic dopamine response and hypodopaminergic dopamine response in the prefrontal cortex, hyperdopamine response in the mesolimbic areas. But what does cannabis do? Well, cannabis, of course, triggers its receptors, the cannabinoid receptors, for example, in the ventral tegmental area. And then you get, of course, this stimulation and this impaired feedback. On the other hand, we know that it also impairs working memory, it can elicit acute symptoms, so all the symptoms of schizophrenia are present if you use cannabis, but what happens if we start using cannabis continuously? So because this is only at one time, and so if you use cannabis during adolescence, how does that explain that you will only develop psychosis, for example, in your mid-20s or something, right? Because this is the age where psychosis onset occurs. Well, we now know that it's not only this contour inactivity, but it is because you have a decreased negative feedback response that you will also increase all your gene expression machinery to have a heightened activity of this VTA mesolimbic system. So there's much more than only this aberrant genetic variant of the CONT allele. We see that this can lead to different expression of all these signaling molecules related to the cannabinoid receptors, and we see indeed disturbances in synaptic plasticity, neural excitability, and also the real network activity of the mesolimbic system is disturbed. Right? And of course this can lead to schizophrenia. One of the examples here is also that there is a distribution, misdistribution between the dopamine and dopamine D2 receptors in the brain. I will not go into detail here today. Now, so to summarize this more or less, so what happens in schizophrenia is, in fact, if you look at the right part of this figure, what we continuously are doing is we receive input from the outside world, from the things that we hear, the things that we see, but mainly from our social contexts. You're looking at me, I'm looking at you. We're not aware of this. It happens completely unconscious, but we are continuously trying to look for emotions on the faces of other people. It's a continuous process, and we cannot stop this, we cannot change this. But why are we not thinking of this continuously? Because we have some kind of negative feedback input from our prefrontal cortical areas. And only when something is really aberrant, then we can see, okay, hey, there is something wrong with this person. And then we can try to reason about this. But normally, these basal inputs that we receive, they come from basal structures like the amygdala and the hippocampus. We call those the bottom-up stimuli. So all the stimuli that you receive and that you're not consciously aware of. On the other hand, you have this inhibition from the prefrontal cortex that hampers the input of these basal bottom-up stimuli. And these signals, they are integrated in the structure, the nucleus accumbens, which is in fact the limbic target of the dopaminergic system. So the mesolimbic system that projects to the nucleus accumbens. So what happens in schizophrenia is in fact that these bottom-up stimuli, they are favored over these prefrontal inhibitory profiles. So we call this a loss of top-down control of bottom-up stimuli, meaning that schizophrenia patients, although it's not some kind of interpretation, because they of course do it from their biological standpoint, they receive all these inputs and they will automatically provide some meaning to this, which is not inhibited by the prefrontal cortical areas, so not inhibited by any kind of reasoning. So, if any of you is looking to me with, well, very, how should I say, like a look that they do not understand me, then I can start thinking, well, why is she thinking this? I'm not doing this right. So there may be a lot of going on in my head, a lot of reasoning that I'm not aware of. But in schizophrenia, you lack this top-down control of all these bottom-up stimuli, and these bottom-up stimuli will take control. And this will lead to positive symptoms like hallucinations and delusions. So it will increase your propensity to develop paranoia ideas, to see things that are not there, to hear voices, and so on. Now, as we have tried to explain in this article, there are indeed all of the genetic variants that have been described in schizophrenia. They, one way or the other, impact on this neural circuit. And the stars here, they indicate a lot of known risk factors that we know if you reduce them to animal models, that we can see that they indeed lead to this disturbed neural network. For example, in animals, during development, if you then make a lesion of the hippocampus, it will automatically lead to a hyperactivity of the mesolimbic system in the nucleus accumbens. The same if you do this with a lesion in the prefrontal cortex. We also know that, for example, having a childhood trauma, like the death of a family or being in a quite scary accident, for example, that sensitizes your amygdala. And this, if you do this also in animals, you will see that it also increases the activity of the mesolimbic dopamine system. So all of these environmental factors, they can cause schizophrenia by disturbing this system. Okay. So for schizophrenia, how are you doing? Are there any questions? No? Is everything clear? I think I will conclude this session here. I had some slides about depression, but I have the idea that this might be too much for you for now. My colleague next week, he will tell you something about epigenetics and depression, and will come back to this part as well. So what I tried to show you is well, how you also can do this gene environment research or genetic research in the context of psychiatric disorders. And it's really well, hardcore molecular biology that we do. For example, in our lab, we're also trying to use cannabis in animal models to try to see what is going on with this dopaminergic circuit in detail. We now know that it sensitizes the dopaminergic system, but we don't know how of which molecular players that are involved there. It has also to do a lot with epigenetic changes and so on. We use also some knockout models or transgenic animals to see if we can introduce some of these aberrant genetic variants that have been found in humans, how the animals respond to this, or how the dopaminergic system is modified by this. So we also have, of course, a lot of animal behavioral analysis. It's very hard to say when an animal is depressed or when it is psychotic, but we have some kind of behavioral tasks that modulate or that model some endophenotypes of these psychiatric disorders. And if you want to know more about this, you can always contact us, and you're free to have a look in the lab and to talk to some of our PhD students or to some of our researchers. And if you want to do an internship with us, you're always welcome to apply for this. Okay? Okay, so I hope to see you sometime.